All right, future doctors, welcome to the Explainer. Let's get right into it. This is a high-energy, rapid-fire review session, and we've designed it to really sharpen your recall and get you feeling prepped and confident for your board exams.
Look, we've all been there, right? You're just feeling completely buried by the sheer volume of information. So today, that's what we're going to do. We're cutting right through all that noise. This is a focused blitz on the absolute must-know, high-yield concepts that are pretty much guaranteed to show up.
Okay, first up, we're going to lay the foundation with some biochemistry bedrock. Because to really understand what goes wrong in disease, you have to master the rules of what goes right first. And we're talking specifically about the gatekeepers that guard our genome and control cell growth.
Now, this one is a classic for a reason. I want you to think of the p53 gene as your cell's chief of security. It's constantly patrolling the genome, and when it finds DNA damage, it slams on the brakes, halting the cell cycle to give it time for repairs. So what happens when p53 isn't activated? Well, it's like firing the guard. This lets damaged cells replicate, which is a huge step toward cancer, and it's what leads to syndromes like Lee-Fraumeni.
So what's really interesting here is how p53 compares to another major player, the retinoblastoma gene, or RB. You absolutely cannot confuse these two on test day. See, p53 is a damage checkpoint. It responds when there's a problem. RB, on the other hand, is like a direct brake on the cell cycle, a constant negative regulator. They're two totally different mechanisms, but both are absolutely crucial for stopping uncontrolled cell growth.
So now that we've seen how these cellular guards work, let's dive into what happens when the genetic code itself gets all scrambled up. In pathology, it is all about pattern recognition. You've got to match the genetic fingerprint to the specific cancer it causes.
You're sitting in your exam. You see this translocation, T.1448. Before you even read the rest of the question, your brain should be screaming one thing:
follicular lymphoma. And the mechanism here is just, well, it's brilliantly simple. BCL2 is a really powerful anti-apoptosis protein. It basically tells cells, don't die. So by moving it from chromosome 18 to the immunoglobulin heavy chain spot on chromosome 14, you're putting it under the control of a promoter that is always switched on in B cells. The result? A massive overexpression of BCL2. Cells that should be undergoing programmed cell death are now essentially immortal. It's the perfect recipe for a malignancy.
You know, these patterns aren't just in the genes. They are so crucial in the clinic, too. So let's use that same pattern recognition skill on a classic point of confusion that trips up so many students: sorting out the major syndromes in kidney disease.
So the crucial point is this: you have got to master the distinction between nephrotic and nephritic syndromes. Just think O for nephrotic. It's all about massive proteinuria in a bland sediment. This huge loss of protein, especially albumin, leads to that classic tetrad: hypoalbuminemia, generalized edema, and hyperlipidemia. Now, on the flip side, think I for nephritic. That's all about inflammation. This means you get an active sediment with hematuria and red blood cell casts, plus you'll see hypertension and oliguria.
Let's power up with some pharmacology. You know, while understanding the mechanisms is king, a killer mnemonic can be the ace up your sleeve on test day. So let's lock in a few of these that are basically guaranteed points.
You absolutely, positively must know the side effects of lithium. But hey, luckily, the drug itself gives you a great mnemonic. Just remember the name lithium. This is going to cover the big ones: low thyroid, heart defects like the Epstein anomaly, insipidus, that's nephrogenic diabetes insipidus, and unwanted movements like a tremor.
Okay, let's actually trace the path of a loop diuretic together. So it targets the real workhorse of the nephron, the thick ascending limb of the loop of Henle. And right there, it blocks that incredibly powerful NAA-K2Cl co-transporter. By shutting down this pump, you prevent all these ions from being reabsorbed. So a ton of solute stays in the tubule, water follows the solute, and that's exactly why these are such potent diuretics.
All right, time to test yourself. Here's how it's going to work. I'll show you the question, you pause and lock in your answer, and then I'll give you the explanation. No peeking. Let's go. Okay. The key here is a CAG trinucleotide repeat on chromosome 4. And you have to remember, it exhibits anticipation, meaning it gets worse with each generation. The answer is the classic 5 T's: truncus arteriosus, transposition, tricuspid atresia, tetralogy of Fowlett, and TAPVR. Just remember, if it starts with a T, think blue baby.
That's going to be human papillomavirus, or HPV. You see coilocytes on a slide, you think HPV. They are absolutely pathognomonic for it.
Hyponatremia.
In a board question, if you see pneumonia plus a low sodium level, that is a massive clue pointing you directly to Legionella.
Okay, the key differentiator is methylmalonic acid. Look, both of them are going to have high homocysteine, but only B12 deficiency will have a high methylmalonic acid. That's your moneymaker right there.
And that's our blitz. You know, from the genome's guardians to the kidneys syndromes, mastering board prep is really all about recognizing patterns. We've covered some of the biggest ones today, but the real question is, which pattern will you master next? Keep connecting those dots. You've got this. Good luck.